All right, it's 8 a.m. Let's get started. Good morning, everyone. Morning. It's so nice to be in a PSA in person, isn't it? It's a great feeling. So welcome to the symposium, Poultry Industry Paradigms, Connecting the Dots. The whole idea behind this symposium is to have a holistic view into different topics that are very important to the global poultry industry, topics that have been definitely influencing different aspects of the industry. We have 6 well-known experts today. They're going to share their experience, they're going to share their perspectives with us today, and hopefully we can learn about and improve our knowledge as well on a wide range of topics, genetic, nutrition, smart farming, processing and meat quality, sustainability and welfare, and market trends. So we will have 4 presentations starting in the morning, then we will have a break at 10 a.m. Then we will come back with 2 more presentations and a Q&A section. Please bring your questions later on today. The Q&A section is at 11:30. If you think you're gonna forget some of your questions from the early on presentations, we have notepads in the back of the room. Please write it down. And if you feel too shy to ask your question, there are sticky notes into it, and then write your question and hand it to me over the break or after the final presentations, and I'll ask your question for you, okay? So with that, let's get started. Welcoming our first speaker, Jesus Orengo from Highline International. He's gonna be talking about genetic revolution. What have we learned so far? Jesus.
Good morning, everybody. Can you hear me okay? Thank you, Anita and Evonik, for organizing this workshop and for the invitation, and thank you, all of you, for being here early in the morning. So what we have learned so far, I think we have learned a lot, and there are many dots into this story, so I hope that I am able to collapse this into a 30-minute presentation. I think for me that was the most challenging thing, thinking about this topic, is how I was going to do it in half an hour. But we'll revisit some of the basic ideas of genetic improvement in poultry, from the application of very basic Mendelian principles to nowadays the full implementation of genomic. Remind ourselves why phenotyping is so important, even in the genomic era. A little bit about genotyping technologies, because I think we are really in the era of the genomic revolution. Selection priorities many years ago and now. I'm not going to speculate much about the future, since this is what we have achieved so far. But maybe we can speculate something in the questions and discussions. When I talk about genetics, I always like to try with a slide similar to this one, to remind myself and everybody that the final performance of the animal, the phenotypes that you measure or we want to improve, being breeding traits or broiler traits in broilers, or any of the production of quality traits in layers, of course, depend on the genetic makeup of the birds. And the genetic makeup of the birds give them the potential, the maximum potential, the biological potential to express all of these phenotypes. But I choose this kind of funnel-shaped illustration to remind that the final performance depends not only on genetics, but the interaction of genetics with many non-genetic factors or environmental factors, such as nutrition and feeding, health, biosecurity, environment, climate, housing, management, etc. And I think it's good that we are going to cover some of these in more detail during the workshop. So I think genetic improvement in poultry is really a successful story. It's a wonderful story to tell and be part of. But I think we can talk about many things today. But one that, for me, it was the key one is when maybe in the 30s or end of the 20s, there was this idea of not doing or not producing dual-purpose birds, but producing lines and selecting and diversifying lines for meat production and egg production, and it really gave a big boost to the broiler industry and the layer industry. So genetic improvement in poultry is complex, but we can summarize it in these figures here. Is the gene flow in the genetic programs. It has a pyramid shape, where we have in the top of these pyramids, we have the pure lines, the elite population or the nucleus population, in which we maximize progress through intense genetic selection. But then we cannot produce enough of this, so we need a multiplication stage both in broilers and layers. And this multiplication stage serves two purposes. One is to multiply the stock, so we have enough commercial birds at the base of the pyramid to produce broiler meat or layers eggs. But at the same time, during this multiplication, there is a crossbreeding process. And this crossbreeding allows us to exploit many other fundamental principles in the breeding program, such as heterosis, combinability, and complementarity, that make this final product really optimal product for the market. So one question I always ask is, why poultry? Are we in the right industry? And I think I don't need to preach to the choir talking about this, but I wanted to summarize a little bit why poultry is so important worldwide nowadays. One is efficiency. I got across this paper, this is aquaculture paper recently, in which the authors summarize efficiency not in terms of the feed conversion ratio, but in terms of calories of protein retention of the final product. And I was not surprised to see that both layers and broilers exceeded the other livestock, ruminants and pigs. But I was really surprised that both for calorie retention in red and for protein retention in pig, that both broilers and layers also exceeded the most popular or the most common aquaculture species. And then in this website and our warning data, we can see a good summary of, in terms of not only efficiency but sustainability. In this graph, it shows the CO2 emissions per kilogram of final product. So in the case of broilers, about 6 kilograms of CO2 per kilogram of broiler, and in the case of layers, 4.5 kilograms of CO2 per kilogram of eggs. And again, it's not surprising that it's much better in poultry than in other livestock, but also many other plant-based products such as coffee and chocolate and oil. And not only in terms of CO2 emission, but also in terms of land utilization. Is this an abysmal difference between ruminants for beef, lamb and beef cattle, and poultry? In this case, 7.1 square meter per 100 grams of meat and 5.7 square meter per 100 grams of eggs. So what is the role of genetics in all of this progress? And the species are efficient biologically and economically, but there has been a lot of effort in selecting for this. And this is data from DEFRA from 2011, and what is summarized here is different species from ruminants for beef and milk, pigs, and then poultry, in terms of what was achieved in 20 years of genetic selection in terms of reduction of this greenhouse emission and global warming potential. And to simplify this data, what I did was to deviate from beef cattle, which is assumed that is the least efficient or the most pollutant in terms of gas emissions and global warming, and put it to zero and then see the deviations. And so it's not surprising that sheep industry is about at the same level, but then there is a significant reduction of about 15% for pigs and dairy. And then for poultry, we have 25 and 23% of reduction in global warming potential relative to beef cattle. And in the case of layer, they surpass all the others for all the parameters here, not only the global warming potential, but also in terms of greenhouse gas emissions. So here in this graph, this is for data from Aviagen that was published yesterday in Poultry World. And I think it summarized the contribution of genetic improvement to the sustainability in the case of broilers from 1970 to now. So in 50 years, they calculated 50% reduction in the carbon footprint of broilers, and they project that 50% additional reduction in the coming 10 years to 2030. Here in the right, in the case of layer, there was a very interesting study. The final report published in the Egg Industry Center, Ohio State, and the full paper published in Poultry Science by Pelletier in 2014, and where they summarized the environmental footprint of the layer industry in the US from 1960 to 2010, so 50 years in the layer industry. And the calculation in that paper showed that the reduction of environmental footprint were quite significant. It were 65% lower acidifying emissions, and 71% reduction in eutrophin and greenhouse gases, and 31% reduction in cumulative energy demand by the system. So both the layers and broilers are making a huge effort in terms of being more efficient and leaving a lower environmental footprint. So I will shift gears now to talk a little bit more into the genetic subject. And every time we talk about genetics, we need to use some formulas. I will try to use not many, because I know that this is not the best audience to talk about statistics, but it helps. So breeding programs started with the application of very basic Mendelian principles in basically measuring traits and ranking animals, relative values, and selecting the better. And in some cases, using family means if the phenotype was not used in the animals. But selection was one trait at a time. It was very slow and complicated when we start trying to improve the birds for different traits. So 1943, Dr. Hazel came with the idea of the selection index, and for me, selection index is the first real paradigm in improvement of animal populations. So the idea is that we have this value H. You can say there is a holistic breeding value, or after Hazel maybe, and so we can maximize and select birds based on this H, which is just a function of different sources of information. This X here, and these different sources of information could be different relatives, or it can be different traits, or traits measuring different scenarios. So it's the first time that we were able to combine different sources of information and account for all the genetic and phenotypic correlations of those sources of information at the same time. And the other thing that was very important, and is very important of the selection index, is that you also could give a weight, an economic weight or a biological weight to each one of these sources of information. And I think selection index stay with us until today. We still select birds today based on a selection index. What it has changed tremendously is how we calculate those X and V's in this equation. Then in the end of the 80s, or mid of 80s, the group of the University of Cornell, led by Dr. Henderson, and then by Quast and Pollack, formalized the idea of the mixed model equations and introduced the best linear unbiased predictor for selecting animals and the animal model. And this was not new for Dr. Henderson. Was playing with these ideas in the 40s, but he couldn't formalize it statistically until the end of the 80s. And for me, this was a very important paradigm shift in genetic selection, because it really transformed the idea of the selection index. So I'm not going to talk much about these equations, but this is a simplified form of the animal model. And what we have here is equations in the top for fixed effects. Fixed effects is everything that is not genetics and we want to adjust in the models. Age, hatch effects, location effects. Many of these effects that we want to account and use random effects are animal effects or permanent animal effects or maternal effects, anything that is random and that we want to predict. So it's a system that allows the simultaneous estimation of fixed effects and prediction of random effects. And what this translates in a nutshell is that we now could predict breeding values to select animals accounted at the same time simultaneously for all of these non-genetic factors. So this was a very powerful tool, also allowed to expand really the portfolio of models that we could implement, from maternal or repeatability model, random regression model, you mentioned it. So the key part of this that makes this really different than the selection index is this G inverse, or this genetic relationship matrix that at the beginning in the animal model accounted for the relationship of the candidate, which was the animal, with the relatives. And then really we entered the molecular era, and I think this is really substantially changed the way breeding programs operate nowadays. And well, you are familiar with the terminology QTL, quantitative trait loci, and that up to this point, I think we are always talking about the genotype and the animal and the improvement of the genetics of the animal as a black box. We cannot see the genotype. We can only infer or predict the genotype based on the information and some smart mathematics and statistics. But when we are able to start identify quantitative trait loci, regions in the genome can be marker of genes that are associated with traits of interest, then it's a completely new scenario. We can start opening this black box and start selecting for specific regions in the genome. And there was this marker-assisted selection idea that we could use these markers or this molecular information to improve and apply selection. And in 2004, was probably also a very important moment for poultry, because it's when the International Chicken Genome Sequence Consortium released the first chicken genome, and it was the first livestock species or farm animal species that had that release, and this opened the opportunity now to explore not few of these markers, but many of these markers. And about the same time, researchers introduced this idea of the genomic selection. And genomic selection is basically a principle in which we can apply selection based on information from many markers. So now we have two tools. One is the possibility of developing many of these markers, and then the statistical framework or the operational framework to utilize that information in selection. And I will talk a little bit more about genomic selection later, because I think it really is a key component of genetic improvement nowadays, not only in poultry, but on livestock and even plant species. So later at the end of the last decade, researchers at the University of Georgia in Athens, but also almost at the same time, researchers in Europe came with the idea of the single-step prediction method. So and it was a way to simplify and improve the utilization of the genetic information for genomic selection. So imagine this gene versus genetic relationship matrix that we use in the animal model or in the mixed model equation, but think that the genetic relationship among animals or candidates or animals in a population has two components. One is the relationship matrix, the pedigree and the genealogy. We know the birth, the parents, the grandparents, the great-grandparents, and so forth. And we have that information that is used in selection in the basic animal model. But then we also have the genotypic information. When we genotype animals and a large number of animals, then we can calculate the genomic relationship. And the principle of the single-step system is that we can integrate that information, combine the relationship information with the genomic information, and create this age matrix, a holistic relationship matrix, I would say, and use it as an inverse in this system of equations. So we're basically talking about mixed model equation of blood, but this time including genotypic information is a G-blob, a genome-based blob. So I think that was an important also advance in terms of how we selected animals. And more recently, and we probably haven't utilized this fully and still in the first stages, is what about instead of using panels of SNP chips or markers, that we could use low-pass whole genome sequence to use that in selection, and that's something that is being used right now, but probably that hasn't been exploited to the maximum potential. So to illustrate a little bit of this summary of the evolution of the utilization of methodologies in the breeding programs, I summarize this with two examples. One is in layers and is based on the experience that Highline has experienced since it was founded in 1936. And one of the first things is that was the first time that hybrids, crossbred birds, were used for commercial production of eggs. And I think that was a very important step. But this evolved rapidly into the incorporation of selection index, or in the 40s and in the 60s, the use of blood types, which now is not that they are markers, but at the time were just biomarkers, but they were used for selecting for different characteristics. So it was the first attempt to use marker-assisted selection already, especially for disease resistance, especially disease such as Marek's. Then I mentioned to you at the beginning the importance of the genotype by environment interactions, and this was very important also to collect information of phenotype not only in the pedigree nucleus, but go to the commercial environments and do applied seed testing or progeny testing, and incorporating phenotypes from challenging environments into the selection program. So on disease resistance, a attends that we started with Marek's disease in the 80s, and we have done since. Dr. Janet Fulton, that is here in the audience, started molecular biology lab in Highline and explored many markers, starting with microsatellites, but later with SNPs. Of course, the implementation of BLOB came in the late 90s, and more recently, full implementation of the genomic selection program. And one thing that the genomic selection, and at least in the case of layers, require is a transformation of the way we manage the population structure. So changing the system from a single farm, discrete selection, one at a time, one generation at a time, all in, all out, to really a system in which we have 3 nucleus pedigree farms in which we have a rotation and continuous improvement, because young males from the young generation are mated to mid-aged females in the older generations, and in that way we can reduce by half the generation interval. And this system, along with the genomic selection, really allows us to accelerate the genetic progress. And I include this because you know about the transformation of the industry and the layer industry in the US and in many countries, that evolving from cage production to cage-free systems, and that requires that the breeding program also adapt to this type of industry trends, because then most of this genetic selection has been done in all of this time using single cage to be able to maximize the information that we can collect in candidates individuals. But now, if we need to select individual hens, but they are housed in aviaries or in cage-free systems, we need new systems like the trap nest, and it's an interesting cycle, because in the early 40s that was the way it was done, using trap nest. Only that at that time it was manual trap nest, and now we are in the era in which there are really mechanical or electronic trap nests. And just to finish this example in layers, 2 things that were really important also for the expansion of the industry was the discovery of genes that allowed feather sexing in the hatcheries to separate males and females, either by color of the feathers in the brown lines, or feather sexing in the white leghorns and in the white lines. And also the eradication of avian leukosis, which you know is a vertically transmitted disease and really can cripple the possibility of a breeding company to expand markets internationally. For an example in broilers of this evolution, I think I present this example from Cobb in poultry side, and we can see similar components of this evolution from the dual purple birds to more specific broiler lines, but they also acknowledge the use of mass selection for growth, and then later selection index for traits like conformation, feed efficiency, and reproductive traits, the introduction of the blood programs and the animal model, and the identification of markers for selection. And nowadays we know that the genomic selection is fully implemented in all the broiler companies worldwide. So I have mentioned genomic selection a couple of times, and for me it's the everyday word, but I want to make sure that everybody understands what is behind this idea of genomic selection and the ways it is applied. So this slide summarizes in a nutshell what genomic selection is. And it's a very complicated process, but it's just basically utilizing 3 sources of information, all from the animals, in the prediction of breeding values and selection. So we capture from layers or broilers, doesn't matter, of cows, sheep, or pigs, 3 sources of information. One is the relationship matrix and the relationship that exists in these animals. We know the birds, the parents, the grandparents, and many generations. And we can use as much of that genealogy information as we want or need. Then we have tissues, and we can extract DNAs and collect genotypes. In this example is a SNP chips from Affymetrix that allows to genotype more than 600,000 markers at the same time for each animal. So having all of this genomic information not only allows to associate each one of these markers with a given economic trait, but also create or build this blue shape here in the middle, which is the genomic relationship matrix. So it's quite dense because every animal that has genotype has shared genotype with other animals in the population. So that's the second source of information. And then all the phenotypes, all the economic traits that we can incorporate, breeder or broilers in meat lines or egg quality, feed efficiency, disease resistance, any trait that we want to incorporate in the selection. So we create this gigantic system of equations, and we require really fast computers to compute all of this information at the same time. And at the epicenter of this, the final output is what is called the genomic expected breeding values. So it's integrating all of this information, in our case with a single step, but it can be a different methodologies, and have a unique only value for each trait that combine all the genotypic relationship and phenotype information from the animals. And as we use that in selection, then it's genomic selection. And as I mentioned earlier, we still apply the selection index, only that the sophistication in which we introduce these X values and those weighting values. So other thing that I really want to say is that genotype is key. I mean, one of the things that I really want to emphasize is the importance of phenotypes. Dr. Lush from Iowa State, which was one of the real founding fathers of breeding and improvement of animal population, used to say that the data is the raw material used by geneticists to improve farm animal populations, and is so true today. When the idea of the genomic selection and the utilization of genomic information came along in the scientific community, there was a lot of speculation that we could tackle very difficult phenotypes that are difficult to measure or measure too late in the life of the animals or in other animals. And we could have good sources of phenotypic information, train models based on markers, train those markers, know exactly what the marker affects, and then apply genomic selection, and then forget about phenotyping, because already you know the relationship between those markers or SNPs in the phenotypes. But there is no that in reality. What we have seen and many researchers have proved is that the only way to maximize the benefits of genomic selection is continuous phenotyping, having good quality phenotypes, and retraining those models again and again and again. Otherwise, that genetic progress eroded quite rapidly. Well, for a 30-minute presentation, it's very difficult to go through breeding goals in layers and broilers. Probably that will be a symposium itself. But I want to remark that phenotypes and breeding goals evolved from very simple recording of body weights and gains in broilers and counting eggs and layers to a very, very sophisticated evaluation program nowadays in both species and also in turkeys. And just to give an example, in our breeding program, if we account for all the traits and measure in different environments and in different ways, we can count more than 30 traits that we use in the breeding program. So you can imagine that we cannot go in detail, but I want to summarize. I really like this publication from Untitled 2013 because it gives a perspective of the evolution of breeding goals, and this is based on the consensus of 21 breeders and geneticists worldwide. So I think it's a good estimation. So this is a polygonal representation of breeding goals from 1950 to 1990 to 2015, and a little bit of projection to 2025. And these polygons in the left and the right is just rotation of the same. So what we can see is that in 1950, there's really one trait that dominated the breeding goal and the selection objective was productivity. And a little bit, there is vector for feed efficiency. So there was a little bit of feed efficiency, a lot of productivity, not much space for other traits, and as selection and breeding goals evolve, and we see now in the 2025, there is not really one trait that really dominates this polygonal representation, and there are still three vectors, main vectors, which is feed efficiency, product quality, and robustness. And notice that productivity went from being overwhelmingly the most important breeding goal to be not the most important one. It's still here, but it's probably as represented as adaptivity or human health or environmental efficiency. And there are other traits that are probably not considered here directly that have to do with behavior and animal welfare that are really relevant nowadays, and that they are occupying a lot of this space too. How are we doing with time, Anita? No, impossible. Okay. Okay. Genotyping evolved really rapidly. I know that this was going to happen. There are low density, mid density, and high density SNPs, and we can talk about that later or in the answering questions. So I want to say something about the customers, how the societal demands have changed because of the incorporation of animal welfare and environmental impact traits, and how this has really made the breeding complex much more complex and the collection of phenotype more diverse, and at the same time needed to use more advanced analytics and genomics. And I was going to give an example of the transition between the cage production and the cage-free, but I don't think time will allow me to do that. But one thing that I want to say and emphasize is that I see a lot of potential, and there's a lot of research going on in advanced molecular and reproductive techniques. I'm talking, for instance, things like gene editing or GMO that has a lot of potential now and in the future, but of course, societal demands, public acceptance, and regulatory pathways are limitations there. I will finish really quickly talking about what we have achieved so far. I think that, we can talk a lot about this, but I have only two quick examples. I think all of you are familiar with this research from Canada and US, using the Athens Canadian random bred crosses, and at the time the commercial 2001. And what has been the transformation of those birds? For market weight at 1.85 kilos, the 2001 birds grow 3 times faster, and you needed 1/3 of the feed. And you can see in terms of traits in body weight, carcass yield, breast yield, and carcass fat that have changed by several folds. So it's a really remarkable story of changes. And in layers, just to have an example, I wrote this summary from Anderson in 2013 and collaborators that summarized the North Carolina random sample layer test and later the layer performance and management test. And this publication summarized more than 50 years and 37 editions of that test in brown and white eggs. And here in green, you can see the difference in terms of sexual maturity, hen day production, accumulated hen house eggs, feed conversion ratio, mortality, egg weight, and body weight. And you can see that for each one of them, there was a significant progress. And something that I will only emphasize because of time is the large reduction on body weight and body size. So that not only that these layers are able to produce much more efficiently with less mortality and more egg mass, but also that they reduce the body weight in about close to 30% in the case of browns and about 20% in the case of white. So this is very important because in layers we have these hens living in the system for about 2 years if there is no molting. And so it's a lot of saving in terms of maintenance requirement for these hens. So it's really remarkable. So to finish, I think the future for genetic improvement in poultry is really bright. We just came this weekend from 1 week in the Netherlands, in Rotterdam, for attending the World Congress of Genetics Applied to Livestock Production. This is the 12th edition of this scientific meeting, and it's incredible. All the evolution that we have in phenotyping and modeling and incorporation of not only genomics, but also all the other omics. So we are moving to the era of the panoomics. So the future is great. But still research is needed to adjust or keep adjusting the improvement to the industry changes and challenges, because the customer evolves every day. And just to finish, for me, I think the thing that will change the needle in the future is the possibility of using reproductive technologies and advanced molecular technologies like gene editing and GMO. So with that, thank you so much, and I'm sorry if I took a little bit of the time. Thank you so much. And I hope that there is something that I didn't cover that we can discuss it during the questions and discussions. Thank you. Are there questions now or at the end? Later. I think questions at the end. Thank you.
Thank you, Dr. Arango. Yes, we will have questions at our Q&A section at 11:30. With that, let's continue with Dr. Phil Smith from Tyson Foods with the presentation, Nutritional Developments, What is Next? And it's almost ready.
Thank you, Anita. I really appreciate that, and thank you for the invitation and for Evonik for sponsoring me to come here. It's fantastic to be in person. Like everybody said, I'm tired of virtual stuff, so anyway, I have a couple of ground rules. One of the things I want you to do is, guys, just relax, because I don't have any high-powered anything really, and it's just what's next is just kind of, what's my opinion. And I ask our team, Roy Brister, Hunter, Rocky, Malia, everybody, to get their ideas as well. So I kind of use some of their ideas as, what's next in nutrition. And that's, so I don't have a crystal ball. So anyway, so relax. And I hope you can remember to ask some questions. I know we're running a little behind already, so I'll try to get started here for anything I can think of. Well, one of the main things I think that's not going to change is working with people. We need to work with our growers. This is a couple of growers in their chicken house. These people grow our birds. We're very sensitive. If we make feed changes, they get paid by growing so many pounds, and it's competitive. So we don't want to do anything to slow those folks down. So being there, working with the producers, actually working with the feed mill hatcheries, the whole team. We need to remember the chickens are our main problem. The people is what we have to deal with, I'm telling you. So your communication skills, getting along with these people. And feed, being 60-something% of the cost, gets a lot of attention from everybody in the company. When things don't go right, it's usually the feed. Maybe the hatchery or the chicks, but a lot of times it's us. So we need to remember these people and communicate. Because I learn a lot from these guys. When I say, how's your birds doing? They're there with them every day, and I'm not there every day. So whether we're in breeders, broilers, pullets, I see Malia back there, she's doing the same thing in the hen farms. And we're also in the meat business. So when I try to think, God put us in a great position. We're producing wholesome food to feed the world. And chicken just saw how efficient it is in eggs and meat. We have such a great story to tell and a great future. I don't see that slowing down. We need all meats. We need fish. We need beef. We need pork. We need sheep. We need all the eggs and milk and everything else. So it's a great honorable. And we got to remember, we're looking at this carcass, a wholesome food that people eat. So we want to keep it safe. So anything we do in nutrition from now on, it's assumed we're going to have safe, wholesome food. No residues, no problems, no salmonella. Glasses. Anyways, I've listed a bunch of things here about sustainability because in the future, everything, for the last 10 years, that word gets a whole lot more. What does that mean? And people tend to focus a lot on the environment. It's all about, my carbon footprint or my water or what much land I'm using. But really, I consider sustainability a whole lot more than that. It's actually, economics. We gotta make a profit, or we won't be in business. We've got to have the environment, and we've got to take care of our people, our social side. Those three pillars is what I think. And formulation can play a role in that. And our goals, how to be more efficient, produce more with less inputs, and then we've got to optimize that to get the best cost, and that includes carcass yield. So we can't, folks. We've got to formulate. That's our goals as nutritionists. We got to be. To me, those are the pillars of being a nutritionist. If you do that, then that's what we're trying to do. We're all trying to compete against each other in the industry around the world. And I'm going to show some pictures of chickens grown around the world, and you'll see a lot of the similarities. Another thing that may change in the future, that's not going to change, but in the future, maybe we're going to go to net energy. I'm not an expert on net energy in poultry, but I know if it could be more precise to characterize our ingredients better, then that's something we may do next. We talked a lot yesterday at the informal about formulating the daily nutrient requirements. Well, you've got males and females, you've got averages of all these things, and how we manage that, formulating precisely to the daily nutrient requirements. You don't have to change the feed every day, right? You've got individual birds, every bird's going to be a little different. We'll try to do more of that in the future. We'll try to be more precise because we don't want to overfeed or underfeed. And that's where maybe using amino acids, we could possibly use, a whole array of amino acids, not basically use methionine, lysine. Sometimes threonine. Those three for sure are used almost constantly. And then we also have valine, isoleucine, tryptophan, arginine, and those could be used. But we've got to have some non-essential amino acids. But by formulating that way, maybe we can reduce our crude protein level. Peter Crystal, guys in Australia have worked really hard on this, and other people as well. We don't want to excrete a bunch of nitrogen. We're going to have we're not going to be completely 100% digestible. So these things may help us in the future. So I see more amino acids probably as they become more cost effective for sure. And computer modeling. We met with Dr. Gauss this morning, our team, a little bit talking about how to use it, comparing, to optimize our operations. And I think that's something that's going to be probably used more in the future. Even if we don't, Dr. Gauss, use your model, we're going to use our own model or some other models to help us, where we can say, how can we produce the most meat? Is it return over, not just return over feed costs, but return over the whole operation? We produce 1 million birds, how much does it cost? How much output do we get? Next is, that focus there mainly on formulation. My next topic is basically around ingredients. Because the two things, formulation and ingredients, is what makes our diets. So what are we going to have in the future? I don't see right off the bat corn and soybean meal going away that much. Those two main things, at least in the United States. Not that they'll always be, they're not, but there's so many million tons and bushels, and that's going to be our core thing. We'll still have other co-products and byproducts like bakery meal, DDGs, all kinds of things like that. I don't see those things going away. Now, some people can use animal proteins. If you can, that's great, because you've got a good animal proteins are good nutritional source of amino acids, minerals, calcium and phosphorus, fat and energy, a lot of good things. So I don't see those things going away. For certain, not completely. But these other novel ingredients that are really, we're already using some of these things, like the insect meal, like the black soldier's fly. There's high protein DDGs, there's seaweed, which may be algae, there's all these things like that may be coming in. If they get cost effective, definitely the industry will use all kinds of different feed additives. If we can get them in large quantities, they'll handle, flow. But right now I'd say like black soldier fly larvae meal is something like maybe $1 or $2 a pound. So that's about the price of chicken, right? So it's going to be hard for those things. But as they get grown more and more, right, Randy, there'll be more of these things used. There's no doubt. And microalgae, I played with it a little bit, single cell proteins, they can definitely be used. How much and how easy, that's to be determined. But I know we could, if they become cost effective, we'll definitely use more of those things. And lastly, this new genetics thing. I'm really extremely high on this. The genetic companies like DuPont or DeKalb or Pioneer Hybrid, those people and a lot of other people, if they could get the corn, we've done this before, high oil corn, high lysine corn, soybean meal, I think soybeans have the better chance, and I'm really excited about that. I think they can take out the raffinose pistachios, maybe reduce the trypsin inhibitor, increase the crude protein, increase the energy. Guess what? You've got a more dense ingredient. If you can get a more dense ingredient, then you need less acres, right? You produce more to feed the world's population with a more dense, and I'm confident with GMO, we just talked about the genetics. Well, even GMO-free, you can still select a bean by bean. You can get higher protein. Now, it's going to have to be identity preserved or we won't be able to capture that value. We just can't mix it with our regular stuff if it's 10% higher in amino acids, and we got to pay more for it. And it's got to be cost effective. So that's an uphill battle because a lot of these things are based off of commodities. They're just basically, corn, soy is a commodity, and they have NOPA trading standards, and they don't really go off amino acid values, right? They're just basically, you buy the bushel or ton, and you get what you get. It's up to you to figure all that out. But I'm confident if we could get the industry to look that way, we could push this as an industry, we could get more. And we don't have a soy crushing facility like Randy does at Purdue, but if we did, and you could get your growers to grow it, then you maybe could capture the value. There's a way to capture value. Oh, niche markets. Now all we're doing today is formulating for all these niche markets, which is okay. 50% of the industry is NAE, no antibiotics ever. There's all kind of other combinations of these things all the way to, free range and organic. So will that happen more in the future? I don't see these things going away. There may be more, even more types of, niche markets. But when you're 50% of the industry, I don't really consider antibiotic-free NAE really a niche market anymore. It's just standard. Do you get anything extra for it? Well, maybe you could get a little, you might could sell a few more places. But anyway, that's something that maybe that will trail off and we'll get using antibiotics again if we need to. So that's to be determined, of course. The future's always changing, so we'll see. But it's all about profitability. If somebody will pay us enough and there's a good enough reason, we can grow, we can do these things. One of the things you got to do is you got to have a third-party audit. You got to have somebody watching over that. If you're using organic ingredients, you got to have those certified by somebody. USDA, if you're NAE, you got to have a PVP process to show that you're doing all this stuff. So, and we do all that. So, just a comment about that. God, feed additives. Oh my goodness! Because of antibiotic replacements, there's so many gut health, enhancing feed conversion, growth rate. Just all kinds. There's a whole array of these. You guys can read them. You don't need me to read them. I'm not against any of these things, so all these things can work. And there's probably a whole list of the others that I haven't even put on here that have tremendous value. And if the cost is right and you need them, they're certainly available. And there's combinations. So absolutely, they're being used. And there's a lot of people in the industry, that's trying to promote these products and rightfully so. I don't see that changing. Maybe they'll consolidate a little bit. There's so many ones, but anyway, it's so broad. It's hard to talk about. Ingredients and finished feed analysis. In the future, what do we need? We talked about that yesterday. We got to characterize these ingredients correctly. And they're going to change every season. You see soybean meal from one year to the next. You may see corn. You may see, things are going to vary. How do you adjust your specifications to capture that change? And then, all these assays, could you get wet chemistry? Do you do NIR? Could you train it on certain ingredients like soy that's consistent? You probably can. On certain other byproducts, not so easy. I'm pretty much a wet chemistry person. I like to do wet chemistry. But even with wet chemistry, there's problems. There's variability. Also, when you look at digestibility and you look at, ileal digestibility, you got AME, TME, you got all these different ways to measure it. And if you did 10 different labs, you probably get 10 different answers, right? So you got to pick what you think and what's relative to you and consistently stay with it. If you jump out there and make a huge change, just be careful. Because the ingredients can change. Be consistent. Try to single source. Try to get from the same supplier, from the same region. Most of the time it's come from the same soy plant. Or if it's come from a DDG plant, I've got 3 or 4. I know those plants. I characterize them. I can use that ingredient. I have no, a safety margin. I feel pretty confident that we'll get it in there right. Regulatory issues. Now, this is another thorny deal. You got the FDA, government agency. They regulate what goes in the feed. So when things are coming in the future, I don't know what, some new thing maybe is created, and if it's safe and efficacious and it helps or improve our efficiency, we need to get it approved. So the government work with the feed industry, the AFIA, National Chicken Council. We need to get those things available, and don't just hold, cost millions of dollars to prove the. But if it's safe and efficacious and has bad value, then let's help them promote the government to approve it. That's something in the future that we don't, the way the government is today, it don't seem like it gets along, so it's hard to get anything done, including the FDA, which shouldn't be a political deal at all, right? It's all about safety, food safety, and that sort of thing. Now, I've got a whole bunch of pictures of different things. Malia's given me some of these pictures from breeder farms. I've got a picture of a breeder farm here. And again, I think being a nutritionist in the future, as like Dave Burnham used to say, you want to be as close as you can get to the birds. If you're in the birds at feeding time, at like 5 a.m. in the morning, you'll see if you got problems. You get a whole lot better perspective than sitting in your office or sleeping in until 8 o'clock. You know, you need to get out there. And I'd say in the future, we need to continue to do that. Don't stop doing that. Because we're producing these hatching eggs, and we want these eggs to hatch and be a good baby chicks. And we want to grow them as best we can. So I got some pictures here of these young chicks on feed and water, and you can see different setups and stuff like that. It's critical. Management's critical. Feed's a big cost, but management, I take average feed and good management and win every time, I guarantee you. So remember that. Feed milling. We talked to I had a guy from K-State yesterday. I went blank, but hey, feed milling is a critical thing. We can put the best formulation together, we can get the best analysis, but we got to get the stuff in the right bins, scale it correctly, mix it correctly, load it out correctly, all the steps through there. We need to be watching that. And then that's regulated by the FDA. So we've got the FDA, we have to be ready for FSMA. We have to be, most same feed mill guys are very, watching OSHA, EPA, all the other, they're great. And it's hard to find good feed mill managers. So as a nutritionist, in the future, don't neglect the feed mill or in the feed mill people. Again, it's the people. And I have a few comments here about just as we talked yesterday, talked about maybe making a finer particle size, make a better pellet. Okay, yeah. I tend to think if we're going to break the pellets up in the way we handle it, I'd rather have a coarse grind. So I tend to want to have a coarse grind, especially in starter feed that's crumbled. I just recommend it. I don't care if it's above 1,000 microns. I'm okay with that. And feeding whole grains, I have no problem with doing that. If you don't have to grind it. If you could take a little bit of whole wheat, milo, grind it through the pellet mill if you're pelleting the feed. I don't like powdery, mixed up feed. So just a comment. I don't care what kind of feed you're making. This is just my personal preference on that. And I told Peter Crystal, I was at one of his farms back in 2010 in New Zealand. On this particular farm, I'm fixing to show a picture of it. You can see the pellets now. They're milo, wheat-based diets. Do you see hardly any fines in that feed? Man, they're making a great pellet there, Peter. I'm telling you. That was at Teagle's in New Zealand, and they're just doing a fantastic job. You saw pellets yesterday, but you won't see much better than that. No corn in that diet hardly at all. Now, I have some pictures from Malia gave me of some real coarse grind, like we're making coarse breeder, and you'll see pieces of chunks of limestone. It needs to flow and handle, but mash feeds and breeders can work really well. We have good luck with that. Just looking at some feed form. Now, I've got a chance to take you around the world for just a second here. We're going to go, this is Hope, Arkansas, one of my complexes. In the middle of summer, it's 100 degrees like it is here today. Believe me, it's hot there. You see all those fans running, and the house is dark. All we're doing is tunneling house. This is just a regular chicken house. I don't remember the age of these broilers, but that's one of our broiler farms here in the United States. This next one is a picture from Taiwan, South Taiwan, way south of Taipei. And it's very double-deck house, very strong. You see, for typhoons come through there and wash the place away. And these people were growing chickens there. This is a few years ago. Similar type house you see, and their birds do really good. Now, we just changed continents. We went to Europe. This is in Italy. What do you see there in Italy, you guys from Europe? You got your window. You got like a clear to let light in, And it's a very robust house, very expensive, very good. Ross, probably 308s, growing the big weights there. They do very well. And again, back to this Teagle farm that Peter showed me. I don't know if Peter's here today or not. Maybe he's gone, or doing something else. But that's in near Christchurch, South Island of New Zealand. That's the outside of that farm. That guy's so clean, man. You could eat off the floor there pretty much. He washes everything down. Everything's just so. And they get world-class performance there. I don't know what the feed conversion is right now, but their growth rate in feed conversion is way better than the United States. And that's inside that house. They had a cable-based system. Again, they were fed that feed I showed you a picture of a little bit ago. So you can get great performance just to show you that around the world, people do things a lot the same. And finally, I have a picture from China. And I don't know if you can see that very well, but the birds are on kind of like a plastic flooring. The manure falls through. Tiered housing can work. In the future, definitely, you don't have to have an anticoccidial. You don't have to. The birds, but you have to be able to get in there and, and service the bird. So if a bird dies, you still got to collect it. But you can get extremely good performance and not have the birds on the floor. So there are some advantages in doing that. And tiered housing, like the Jansen house. Whether we'll do that gives you a chance. If you go vertical, you could have 3 or 4 tiers. In this case, this is just one single level. And there's I didn't take this picture. I haven't been to China. Matilda Alfonso gave me that picture. Very much a yellow-legged bird there. They're breeding corn there. The other thing, just to sum up on basically, is the importance of having your research facilities. And I can't emphasize this enough. If you don't have one, have somebody at a university or somebody that's doing private research that you can collaborate with, because all these products are coming. You need to know how to use them. If you have your own research facilities, and pens like we have here, you can have batteries, I guess. You can do different ways of doing research, absolutely. But if it's your own birds and your own pens and your own feed in your own environment, you have a lot better understanding. People bring me research all the time. I have to sift through all this, right? As a nutritionist of the future will have the same problem. They need to figure out what of these things actually are cost-effective and work in their operation at their time. And sometimes I'll see something that works really good. Frustration is I might do it 2 or 3 times and it looks pretty good. The 3rd or 4th time it might not show so good. The consistency is important. So it's really critical, I think. And the problem with that is the companies don't want to spend the money for research is expensive. It takes time. It takes effort for the nutritionist. But I really strongly believe in that. I don't see that going away. I think there's fewer and fewer universities doing research. So it's harder and harder to get outside research done. So I'm very partial to that. So in conclusion, what I want to just leave you with brightness is that the industry's in great shape. We're making money. There's lots of challenges, of course, of COVID, labor. There's shortages. Getting trucks and getting ingredients is the hardest thing in the world right now. It's terrible. But chicken prices are good. So we're overcoming that. Now, but there'll be rough times after that. Chicken market, we're still, even with that, you're never making enough money. I can tell you from my own experience, if you're making a lot of money, you're not making enough. You got to make more. So, and that's good. We should be challenging, always try to improve. So what I'd say is that, the industry's going to continue to evolve. There'll be things I don't have any idea that's going to happen. We had avian influenza really bad. There'll be diseases. There'll be other droughts and things like that. We'll have to adapt, right? We're going to have to move and use the ingredients we have. So anyways, I think that's the last slide, and I thank you for your attention. I hope I got us sort of back on time.
Thank you, Dr. Smith, for the educational and inspiring presentation. We're really on time. This is great. Let's now welcome our 3rd speaker, Dr. Lilong from the University of Georgia, which will give us the presentation Developing Next-Gen Data Management Strategies for Smart Poultry Farms. Just 1 minute, I'll open your presentation. I'll hand it over to you. Just a bit slow. I think you had pretty good pictures there, didn't you?
Try to upload online today.
No worries. coming through. There we go. I'll get that for you.
Thank you. Good morning, everyone. Thank you, Anita, for the invitation. My name is Lilong. I'm assistant professor and engineering specialist with UGA Poultry Science Department. I'm glad to talk about developing next generation of data management strategies for precision farms. Poultry production plays a very important role in feeding increasing water population. Currently, USA is the world's largest broiler producer and second largest egg producer. Because of continuous innovation in animal breeding, nutrition management, environmental control, and disease prevention. But our production still faces a lot of challenges, like the AI outbreak has led to the depopulation of 40 million birds in the US so far, and environmental impact like greenhouse gas, ammonia emissions, and animal welfare concern. The fast-growing broiler chicken tend to have leg issues and heart failures. Not all the birds, but 20–30% of birds have laminitis issue by the end of the flock. Then to trigger the animal rights group to push our production go back to slow-growing chicken, like 50–60 years ago. But don't forget we have 9.5 billion people to feed by 2050. We cannot afford to sacrifice the production efficiency. Instead, we have to look at a new method, new technologies, like precision poultry farming, PPF. PPF is using advanced sensors, sensing technologies to do the real-time data collection, analysis, interpretation for optimizing production, reproduction, animal welfare protection, animal environmental management. A lot of people think PPF is a new concept, just came up in recent years. Actually, we have been working on them for decades. We should give credit to the people who contributed to the environmental control, like heating, cooling, air quality management. That's why our chickens can grow so fast today, from January to December. We can call that technology the first generation of PPF. Nowadays, we are working on the second generation PPF. Besides production environmental management, we also look at animal welfare, food quality safety, and some others like energy efficiency. The key technology and data management strategies we're using for PPF 2.0 is AI technologies like deep learning algorithm that can be used to analyze the videos, images, and audio sound of chickens for predicting their welfare health. So in today's presentation, I will introduce both generations of PPF technologies and data management strategies briefly. For PPF 1.0, environmental data management. Back to 20 years ago, National Academy of Science released a report said animal production like swine, poultry, dairy, beef contribute a lot to the environmental impact like greenhouse gas, ammonia emissions. So we should regulate that. EPA and the Agriculture Research Council worked together and they raised $50 million and selected Purdue University Agriculture Air Quality Lab to select a new project called National Air Emission Monitoring Study, NAMS project. We'd like to develop a national emission baseline and emission factors. The project lasted for 3 years. It cost $14.6 million. That project covered broiler, layer, swan, and dairy. Purdue University couldn't spend all the money, so they gave subawards to 8 other universities from East Coast to West, like Cornell University in New York and UC Davis in California. And I was studying at Purdue University at the time. The PI, Dr. Al Heber, appointed me to lead the monitoring of Indiana layer farm and data analysis, and also co-leading the data analysis for North Carolina poultry farm,
swine farm, and California dairy farm, and Washington farm. I will introduce poultry farm monitoring in today's presentation. For poultry production, no matter layer or broiler, the primary emission sources include poultry harvesting, manure storage, and the land application, because chicken manure is very good soil amendment. Among the three, poultry harvesting is the primary contributor of air pollutant, because fresh manure contains a very high amount of moisture, like 75%. Then it could accelerate the decomposition of uric acid as urea at first, then hydrolysis as ammonium nitrate, and then ammonia. So water plays a very important role in that. That's why NAMS projects focused on the harvesting emissions for both poultry house and the swine house. This is a emission calculation equation. We use this simple equation, ER emission rate equal to ventilation rate Q times concentration difference in the outlet and the inlet. Because outlet air has a higher concentration of air pollutant, and the inlet is currently fresh air. So what we did for NAMS project was quantify each individual fan's running speed, running time, and the performance change over time, so we can quantify the fan's ventilation rate. And then time concentration of air pollutant. For air pollutant, we sampled air from outlet and inlet by using gas sampling system. We bring the air from like 500 feet away to the lab, and we calculated that. That took 5 minutes. So we sampled 10 minutes for each location, and we use only last 3 minutes data as accurate data to do the emission calculation. In the mobile lab, we have many advanced sensors, like we use the Innova to monitor the ammonia greenhouse gas concentrations. Each equipment costs about $50,000. The NAMS project purchased 30 Innovas. I will use the Indiana layer farm as example here to introduce how did we quantify emissions. The farm had 8 chicken houses, layer houses. Each house had 250 layers. It's a manure belt house. The house was ventilated with 88 exhaust fans. In summertime, all the fans were running. In wintertime, only limited number of fans because producer afraid of cold stress, because Indiana had a very cold wintertime. The house had 6 variable speed fans, fan 10 south north, fan 21 south north, 33 south north, and the fan had 3 speeds. In cold winter, it was running the lowest speed. Only 6 fans was running. So you can imagine the air quality should be not good. And for each fan, we installed a vibration sensor to monitor running time. Because if fans running, the fan motor would be vibrating, so we can get a signal 1. If the fan stopped, we get signal 0. So over time, we can quantify the running time. For fan running speed, we use anemometer for each fan, because fans running speed are changing over time, because indoor and outdoor has pressure difference. And also, over time, the dust accumulation on fan blade, damage on fan core, and a motor damage and a slippery in the belt could also affect fan's performance. So we have to keep monitor the fan. We did that work for 2 years. In Indiana layer farm, we used about 300 sensors, and all the sensors was connected to the National Instrument Data Acquisition System. And my Purdue co-advisor, Dr. Ji developed this software based on LabVIEW called AirDAC, Air Quality Data Acquisition and Control System. This system was one of the most powerful in the world. Then it could not only collect 300 sensors data in real time, but also convert that to a graph. So by looking at the graph, we know if all the environmental conditions are in good condition, like ammonia concentration, CO2, and if all the fans are running in good condition. And more importantly, we can remotely access all the site computer in different states, like in New York, California, in our Indiana office. We can do the remote equipment calibration, because an ammonia sensor, for example, should be calibrated weekly Otherwise, the performance may be not accurate. We have to check that. By remotely access the computer, we just click the button, calibration. The side gas cylinder will be wrong, will be open automatically, so we can do the sensor calibration. Over here, I will use ammonia data as an example to introduce how did we quantify emissions. The above graph is ammonia level. The red line is barn A, the blue line barn B. The two houses had a similar ammonia level over 2 years, but differently sometimes. Barn A had the lower concentration because of molting. So farm operation could affect air quality. And in general, the two-house ammonia level was corresponding well to the ambient temperature. Inversely, because if outdoor temperature is cold, like in winter 2009, January 2009, the ambient temperature was very cold, lower than 20 Celsius degree. So the farm had to reduce the ventilation because it really afraid of cold stress. On a cold stress, the bird will eat more feed to maintain the body temperature. That's not good for the economy. So they reduced the ventilation. But the bad thing is air quality. The dust, ammonia level all went up. For the ammonia in that time went to very high. NIOSH, National Institute of Occupation Safety and Health, recommended to use 25 ppm as health line over 8 hours. If about 25 over 8 hours on average, our health and animals' health will be impacted. From this graph, you can see that winter the ammonia level was about doubled, reached 50, 60 ppm. So we know the birds are not healthy. Their welfare was suffering. And the EPA is currently analyzing our data for improving the Clean Air Act. And use the air quality data plus the ventilation data, we quantified that Manubel house had the emission rate of 0.28 gram ammonia per bird per day. That is the Midwest situation in Indiana. In the southeast, like Georgia, we have like warmer climate. The air is more humid, so the emission rate is expected to be higher. If we just use the 0.28 times the total layer numbers in the US, 330 million, we can get the national emission inventory for 1 day. That's close to 90 metric ton, very high number. So we have to consider mitigation. That's why our studies provide a basis for the climate smart poultry farming. That belongs to the second generation of PPF. For the second generation PPF, because of time reason, I will just introduce machine vision and image data management. Prior to introduce my machine vision technology, I would like to mention some pioneers. Dr. Daniel Berkman in Belgium, who developed dynamic system, and Dr. Maureen Dawkins in the UK, who developed optical flow method. Both dynamic system and optical flow method are using machine vision technology, image analysis differently. Dynamics is focused on the pixel change rate over time, and the optic flow is looking at pixel displacement and motion change over time. The left picture is an example of dynamic system. They put bird with different gas score in 6 different pens. Gas score is indicator of animal walking ability. Gas score 0 means bird has a very healthy leg, no problem at all. Gas score 5 means bird has severe leg issue, like dead bird, couldn't move. So theoretically, it's correct. The bird with healthy legs is more active, they run faster, locomotion is higher, so the pixel change rate in the imager is higher. Based on that pixel change, we can predict, okay, the gas score should be higher, or should be lower. The bird has no laminate issues. But in commercial production system, there are so many other reasons can affect bird's activities, like light intensity. If the light intensity is higher, too high, the bird may be more active. And the temperature, very easy to understand. On the cold stress and the heat stress, the bird performance are different. And also the leaf quality can affect bird performance as well. If the leaf is too wet, the bird will tend to have foot pad lesion, dermatitis. So that's not leg issue, it's foot pad issues. That's why currently this kind of system has high uncertainty. And currently no system is available for commercial chicken house to use for tracking individual birds. But for the individual tracking, that's so important. If we cannot track the bird, how can we identify the bird with leg issues for treatment? And why so many scientists are currently working on that. The same by my team at UGA. Currently, we are working on the individual broiler tracking. By doing that, we divided the floor pan into different zones because bird performance and activities are changing from one zone to another. For example, the bird is eating in the feeding zone now. After a couple seconds, the bird would go to drink water because a part feed requires 2 parts water to digest. And a bird will go back to eat after drinking. So by monitoring the back falls between feed and drink, we can track the bird walking and the footsteps, so we can tell the gas score and laminates or not. One bird is easy. How about 20,000 birds in a commercial house? It's a big change. So we start from 20 birds. My student developed a BP neural network. Then it can track 20 birds at the same time. We can tell the real-time number of birds and feeder, drinker, and we can quantify the locomotion. But the early model has some issues, like if the bird huddled together, we will lose the track. We cannot tell which one is which. And also the model has a relative slow running speed. That requires like CPU computer to run. And currently I'm working with a group of computer scientists at UGA to innovate a deep learning model. We developed a different kind of deep learning model, and finally we tested some with very good performance, like a CAN conventional network model, like ULV series model, TensorFlow, Keras. That works very good in individual bird tracking. We are not developing a model from scratch. Instead, we borrow idea from a computer world, like Google, Amazon, Oracle. They have open source database we can use. Like Google is developing self-driving vehicle, and their model is open, free for everybody to use. So we just integrated image of chickens and other information like laminates, walking step, so we can run the model. And my students spend a lot of time on the model training, validation, and test. This is a short video about the bird tracking. We can track 50, 60 birds at a time, no problem. But when the bird walk beneath the feeder water line, sometimes we will lose the tracking. So that's why my student developed a method for reconstructing images. For example, the left picture shows the bird was blocked by a feeder drinking line. We can only extract a part of the images. By reconstructing the image, we can tell, okay, that is a chicken. We don't have to recover 100% chicken image, but we just need to tell that's a chicken, so we can continue to track the bird. We can quantify the daily moving track locomotion, how many miles the bird was running. That model method works good for large bird, because large bird is bigger, not easy to be blocked by equipment. But for small bird younger than 1 week, sometimes the 100 birds was covered by the equipment. Maybe a mobile system is a solution. We can collect the image from different angles, so we can improve our tracking ability. That's broiler chicken. We also applied the imaging technology for cage-free laying hens monitoring. Yeah, in the US, we know that the egg production is shifting from a conventional cage to cage-free because animal welfare concern. McDonald's and Walmart have pledged to buy cage-free eggs only by 2025. But the question is, does cage-free guarantee a bad animal welfare? Maybe not, according to our studies. Cage-free has a lot of issues, like high mortality, injury rate, like Cuban damages, very important issue, packing damages, and also flow eggs. That's leading to the high cost of labor costs. So my students are using imaging technology and other methods trying to address each of individual issues now. Like we can use the imaging system to monitor perching behavior of bird. So we can tell if the bird has leg issues, if a bird has severe Cuban issues, because the bird with severe Cuban damage may select to perch on a lower perch, not higher. So we can quantify the number of birds on different heights of perch, so we can tell which group bird has better Cuban, which bird has a poor Cuban. We are still working on that. And also we can monitor the packing behavior of the bird. I talked with the Midwest from a lot of the packing is an important issue. That's the one primary reason leading to high mortality. If one bird is packed, if the other birds see the blood, they will all go there to pack. By address that issue, my student developed a deep learning model that can track the packing behaviors. So from this short video, we're 80% confident that a bird is packed by other birds. And also, we can quantify the number of packing over time. For example, if some chicken house has higher packing number than others, we should suggest the farm staff to check what is going on. Because packing is caused by some reasons. But we found that light intensity, color can affect packing behaviors. And also air quality can affect packing behavior as well. Especially in wintertime when we reduce ventilation, the dust level is high, ammonia level high, the bird maybe feel uncomfortable. In that kind of environment, they tend to pack more. And also, we can think about other prevention technology method. Packing is natural behavior, and we are not recommended to do big trimming nowadays. So we can use other solution like packing stone. Instead of packing on bird, we can encourage birds packing on block stone. They can get some minerals. And also, the block can be made from like gray material. The bird, by packing, the bird can get some gray material. They like that. That's fit. And also, we notice bird has preference on the color of packing block. So in the future, we would like to develop like SOP for commercial cage-free producer to refer for preventing the packing behavior so we can improve the animal welfare. No matter packing or perching, we have to do individual tracking. By tracking them, we have to innovate our models continuously. We can currently track like 100, 150 birds on the floor. But in commercial house, there are 1,000 birds, like a big Dutchman system, mathematics, they have different number in each session. We have to keep innovating our models so we can track 1,000 birds at the same time. Besides individual bird, we also track floor eggs, because egg laid on the floor could be contaminated by some E. coli, and that's important food safety concern. We can track the egg and let the farm staff know, okay, you have so many eggs over there, you have to collect. If not, other bird would think that is a nesting box. They would go there to lay eggs over there. And besides, the floor eggs are laid for some reasons. We found that the light intensity, light quality can affect floor eggs. For example, in Midwest farm, we recommend the staff to install some light beneath the aviary system where it has a very dark environment. By doing that, they can reduce the floor eggs by 80%. Very impressive. Besides 2D camera, we also use a 3D camera, thermal imagers. This is an example of thermal imaging technology for heat stress evaluation in a commercial cage-free house. You know, in the Midwest, most farms don't have evaporation cooling system because their primary concern is cold stress in wintertime. But the birds are suffering in summertime. So I could be the first to introduce the sprinkling system for the Midwest cage-free house. But in summer, we sprinkle the water for 20 seconds. Don't spray too long, because if too long, the later flow would be wetted too much. So then it won't recover in 1 hour or two. 20, 30 seconds should be good. By doing that, the bird surface body temperature could be reduced by 12 Fahrenheit degree. That's very good. And the farmer has installed the sprinkler system for their other new chicken houses because they noticed that's a method for reduced heat stress. And by looking at a deep picture, the bird was huddling after sprinkling because a little bit of cold dripping water from the sprinkler. The bird noticed that the water is cold. They wanted cold water. So that's a good information for informing the farm manager to flush the water line. If you don't have the sprinkler system, you can flush the water line periodically, so the bird can drink cold water. They can reduce the heat stress. Currently, we are using FLIR Tour, a default software from the summer camera, but that is a manual method. My students and I are working on the method. We're developing automatic tools. We can extract thermal imagery information automatically. So in future, we can predict the body temperature of bird automatically about one picture. So so we can improve the data analysis and animal welfare evaluation. In long term, we would like to integrate all the individual 2D, 3D thermal imaging technology as integrated system. We can install the system in the chicken house for mobile scanning. For the commercial broiler house, for example, is 500 feet long and 50 feet wide. We don't want to use 100 cameras. That's too expensive, and it's harder to collect data, analyze data. Big data doesn't mean good. We need a smart data collection. So use 1 or 2 camera. By using mobile system like chicken boy system or poultry hawk system, they have developed the moving system. By installing our camera and system, we can do the periodically scanning of the house. Like hourly, we can scan the chicken house. We can collect data and store this data in the side computer. The key part is data analysis and deep learning model. We need a fast GPU computer. We need a very good deep learning model. Instead, we can do the cloud computing. Currently, I'm working with Orengo company on the cloud computing. By uploading our image videos to their database, we can use their supercomputer. So then it could accelerate our data analysis greatly. And eventually, we would like to develop an app for producers to use. A producer, you don't want to expect them to learn computer science. They prefer to sell the farm instead of learning so many computer science knowledge. And they like app. With smartphone, they can just click the button, they check, okay, chicken house has the more leg issues than chicken 3. What's the reason? They can set stuff to check what is going on. And for the mobile system, we have GPS. We can tell where the bird is, so we can identify the bird quickly for management. So in that way, we can improve the general house welfare. And data can be used by other stakeholders as well, like Walmart, McDonald's, you can use the information. In summary, data science, like deep learning model algorithm, is the key for precision and smart farming. And the precision farming requires the collaboration between producers, animal scientists, and the computer scientists, and all of us, because each of us has a unique background. Producers know their chickens, like farmers work with the chickens every day. They know better than any others of their chicken. And AI people, they know computers, but they don't know chicken. So I'm an egg engineer. I stand between the animal people and computer people. I'm a kind of technology translator. My job is to promote the collaboration, to make the two sides understand each other better, so we can improve the work. And no matter precision of climate smart farming, mitigation technology, imagers, that could be very expensive. We should not expect a producer to pay everything. Instead, we should encourage the government to consider offset. And also grocers, Walmart, McDonald's, if you wanted to sell the welfare protected product, are you going to pay 20%, 30% higher price for that? That should be reasonable. Otherwise, farmers are not willing to do the work. In past 5 years, my team has published about 25, 30 publications related to precision farming, smart poultry farming. If you like any PDF copy, feel free to let me know. And I greatly appreciate my lab members. I used to have 5 students who just graduated through over here, Ramesh, Shaw, Sachi, and I also appreciate my collaborators. Besides our poultry science colleagues, I worked a lot with computer science and engineers at the UGA. And also I appreciate my sponsors. Thank you very much.
Thank you so much, Dr. Chai, for addressing such interesting and evolving topic. With that, let's welcome our next speaker, Dr. Case Owens from the University of Arkansas, with the presentation, Poultry Processing and Meat Quality, Challenges and Outlook.
Okay, I know I'm a little shorter than the other speakers, so hopefully you can see me over the podium. But thank you for the invitation, Anita, to participate in this symposium. It's so great to be back in person and to speak to such a prestigious group, because you know I love my poultry peeps. So I'm going to talk to you about processing. That's kind of my gig. So I always like to start off in my classes and different conversations I'm talking about per capita consumption of poultry versus other species and so forth. And I've shown this graph quite a bit over the years. And in preparing for this talk, I looked at it a little bit more, and I'm like, you know, we should really be proud of how much growth that we've had in the industry. So the broiler production has just really skyrocketed over the years. Turkeys have kind of remained steady, but we had a little increase in the 80s and 90s. But I put this in there not only to show that growth, but also to tell you that I'm going to concentrate a lot more on the broiler side throughout this presentation, though some of these things that I'll talk about can be applicable into both turkeys and broilers. So broiler market trends, again, production has more than doubled in 30 years. That's a lot of production. We're producing nearly 1 billion pounds of product per week, and that's a lot of poundage. So this increase can be attributed to just increasing broiler size and meat yields. So we have different genetic strains that have high breast meat yielding birds. So you saw some of those pictures in the first presentation by Jesus, which I appreciated. So we have a lot of breast meat yields. The U.S. consumers have really preferred that cut of meat for a number of years, and can give us a lot more products. I'll kind of show some of that development. We've also had improved performance, as that has been discussed in the previous presentations as well. And then in talking to consumers, they're like, why do you have such big chickens? You're breeding these big chickens. I always need to remind people that we're growing them longer also. So, you know, they are growing fast, but with the duration of growth is also increased, and so that results in to a big bird. So the graphic that I have up here is really to show that, hey, we have an increased demand and production, but we also have various market segments. I have a pie graph that I normally give, but I'm going to kind of divide it into these 4 groups. That we have a small bird, a medium-sized bird, and then some big birds in the market. In more recent times, our market has increased to where over half of our production are 6 pounds and greater. And that's going to lead into some of the problems I'll discuss in a little bit. But now over 70% of our meat, or nearly 70% of our meat that's produced, is coming from birds that are 6 pounds and above, or that 2.7 kilogram mark. So to look at the historical developments in processing, I kind of put this together. I thought it was a little fun graphic. We start with just the initial stages of our industry, really a whole bird market. Whole birds were the primary product. And then we moved into parts. All right, so we start cutting that bird up. Consumers are willing to pay for that. Boneless breast meat in the 80s or so, I guess. And then once we produce that boneless breast meat, that could lead into a lot more areas of further processing, where we could produce as an industry further processed products like a deli loaf, a chicken nugget, things of that can be provided to consumers on a ready-to-eat basis almost. And then I've got this chicken sandwich and some different parts at the very end of this, because we do produce those large broilers. They have a really large breast fillet. And we don't just eat the large breast fillet on our plate. We do a considerable amount of portioning, and that really feeds into those fast food markets. So if we want to look at the kind of research that's come along the way, I may need to put my glasses on to see this from here, but automation was probably one of the first things. So the industry we're proud of is that we're really automated, especially on the broiler side, for processing, getting that bird from the live bird to the ready to cook carcass. So automation is definitely key in that development of processing. With the increased demand of boneless breast meat, we needed to look at deboning processes. There is biochemical processes that are going on in the muscle that where if you debone early, you can toughen the meat. And so some of the things that we researched back in the late 80s and early 90s was electrical stimulation. That's postmortem electrical stimulation. I'm getting ahead of myself. Electrical stunning and controlled atmosphere stunning was really starting to be researched in that 80s and 90s period, and we're still continuing that research along the way. There's been some good presentations on that this week as well. So those stunning parameters are also going to come into play later I don't talk about it too much during the rest of the presentation, but as consumers want to know more about their product and welfare, there is an interest in that controlled atmosphere stunning. All right, here's optimized deboning times for quality. So I did get ahead of myself a minute ago. As I said, if you debone early, you get tough meat. And so what was the optimum period of time? There's good research in the 80s to show those tenderness curves that could develop. Hey, if you wait to 24 hours, you get a really nice, tender product. But do we have 24 hours to wait if we're really trying to put that product out? So electrical stimulation used in the postmortem period was developed in efforts to hasten that rigor development so that we could debone earlier without negatively affecting tenderness. So that was kind of the start of my graduate school studies, was electrical stimulation back in the mid-90s, but it had been going on for probably a decade before that. That process is now widely used in the industry, and there's still some tweaks to work out, and I think some research to be done in that area, especially as we're processing such large birds. Is electrical stimulation done in the same manner in a small bird as a large bird? Do you get the same effects? All right, so when we started seeing that increase in deli meats and things of that nature, we had an issue of pale, soft, and exudative PSE type meat, especially with turkeys, but we saw a little bit of it in broilers. That kind of reared its head in the 90s. That was also part of some of my graduate work back in the day. And you know, if you talk to the industry, they're saying, well, we don't have those problems, but I will tell you that I still get calls, usually in the summer months, say, oh, my yields are kind of down. So I think that there is still a little bit of continuum. There's probably some progress that we've made in those areas, but it can rear its head during those heat stress months. So marination research also was kind of taking off in this area, because to make a deli loaf, you have to be able to marinate that product and manipulate the proteins to allow them to bind into a cooked product and stay in a bound manner, and to improve that quality. So there's always research that's going on to improve that functionality and so forth. So more recent time, portioning, vision systems were created in the last 20, 25 years. X-ray for hazards like bone or other foreign material. And then current issues are myopathies. And I'll kind of go through some things that I think are kind of hot topics for the future as well. And I don't want to forget food safety. There's been a lot of food safety work over the years. You know, the implementation of HACCP a little over 20 years ago has kind of changed things as well in the processing world. We've seen a lot more antimicrobial usage, online reprocessing, and a multitude of other things. That's kind of been ongoing throughout the process. So what are our concerns in the marketplace now? Really, it's labor shortages. So our automation really kind of lends itself to those things, but we do so much deboning and further processing. We need to continue with automation in those areas. So there's really a high turnover rate of the labor force due to a variety of things. One, the environment. If you've been in a plant, there's really cold areas in the plant that can be difficult to work in. Sometimes there's really hot areas in the plant. I kind of feel like this room is a little meat locker today, but imagine working in, you know, kind of that 45-degree environment all the time. Repetitive and tedious tasks. So if you're on a debone line, you're doing that all day for an 8-hour shift. And then, of course, the pandemic. That's created a lot of other challenges. So there is definitely some labor issues associated with things today in processing. I've met with a lot of kind of restaurant chains in recent years, and they're really concerned about the supply of product that meets their customers, their specifications of those products. And I'm going to get ahead of myself a little bit, but because quality is, I feel like, a bigger concern today, they are more concerned about their specifications. So size and quality. So we are doing a lot more portioning, but if we're portioning that 9-pound bird to get a chicken sandwich, is that chicken sandwich the same quality as a small bird, meat that comes from a small bird? So those are some things that they're concerned about, like, hey, I've got to change and I've got to go to a larger bird. How is it going to affect the quality of my products so that my consumers won't notice a change or whatnot? Quality overall, I appreciated some of the information that Jesus shared earlier about genetic selection and so forth, that especially that graph he had in the end where we have a very, very production oriented selection in past years, but I really appreciated that product quality was kind of making its headway in more recent times, because it does take a systems approach. The quality of the chicken that we're producing does not just happen from what we're doing in the plants. There's a lot of live production effects that it can impact processing or impact that quality, and so we really need to be working with kind of that overall system to approach those issues. And then food safety is also always a concern, and I'll kind of go over that as well. So in areas of research and development processing, if you look at the quality area, quality can be impacted by production effects, so things from the live bird. That would be like, I'll talk about myopathies in a minute. Processing impacts, all right, so that early deboning or electrical stimulation, those things can impact the quality of that product just from those processing techniques. And then there's further processing technologies. I'm not really going to talk about that a whole lot today, but there's definitely a lot of areas of research and development in that aspect. So we all know that those genetic and growth rates are changing. It's not the same bird. The genetic potential is there, but what kind of trickles down to the ends. There's a lot of things that can impact, from the nutritional side, the management side, that can lead into some problems that we've been observing in recent years. So in the last decade or so, we've really come across these breast myopathies. I'm sure you're all aware of them. They're kind of been named kind of growth-related myopathies. These are myopathies that do show histological lesions where we have fiber degeneration, fibrosis in the muscle, lipid infiltration, inflammatory cells, and et cetera. There's been studies out there, lots of studies about myopathies, but there was a study out of Italy that showed that all of our birds have the potential to have some of these histological changes in them. And then when we get to a certain number of fibers being affected, that's when we see more of those lesions kind of from a visual standpoint. So some of the myopathies that we really are concerned with are white striping, woody breast, and spaghetti. There's a bunch of research out there. Kind of like I said, this is where a lot of the research community came together to really look at what's going on with the bird. Look, I'm a processing person. I'm not a molecular person, but I hope that I've been able to either share information or collaborate with people that can help solve some of these issues. There's a lot of people in the room that are a lot smarter than I am in those areas. Excuse me. There was a great review paper by Zambinelli in 2017 that showed some of the potential causes. Who knows what I did with my glasses, but basically we've read for these larger broilers leading to larger fiber diameter. That leads to issues with oxidative stress in the muscle, so it's kind of an ongoing theme of what's going on. We've got upregulated genes that are associated with oxidative stress. That can then in turn affect altered glucose metabolism and lead to fibrosis and also fiber degeneration. Some of this is related to inadequate vascularization. Satellite cells have been studied. Have those been able to function properly? Some of the research shows that you have to have adequate vascularization for those to be effective and able for muscle repair. So there's a lot of causes out there. I think there's still a lot of research going on in these areas to look at genes that are associated with these things. So I don't think we have the solution yet, but we're, I think, making headway with the genetic companies and so forth. But I kind of want to show what white striping, and you're probably familiar with this, is that white striping does increase in severity as the birds generally get bigger, especially with the higher breast yielding strains. Although you do see this across all different strains, and the lines I have in these graphs are really just for demonstration, but we have high breast yielding strains in these and standard breast yielding lines that are represented in this data that's shown. But it's just showing an overall increase in white striping as the bird size increases. Woody breast, the same, is that it increases as the bird gets larger, and also as the breast yield increases. So the graph of the two graphs I've shown here just are about body weight, but the high breast yielding strains generally have more woody breasts than the standard breast yielding lines, but that definitely increases as the birds get bigger. And as I said, most of our birds, or over half of our birds that we're producing, are over 6 pounds. And so after you get over 6 pounds, that number can really start increasing, or has the potential to increase. So I collected a lot of this data from 2016 to 2018 or 19. The pandemic hit. I haven't been in a whole lot of plants in the past 2 years to see how much of an improvement we've had. But in discussing with some processors, they say that their incidence is a little bit lower, which is a good thing. And some of that may be from genetics, but also from nutritional and management strategies. Spaghetti breast or spaghetti meat is also kind of rearing its head more so, I think, in the last few years. So this is a condition, if you're not aware of it, it's just very soft and fibrous. I put it in the same categories of these other myopathies. One thing that we have noticed is that we see this problem peak a little bit earlier. So in that 5 to 6 pound range, it's kind of in a high area. And then as the birds start getting bigger, that incidence goes down. But you can see that the third picture that I'm showing up here is really from a large fillet, that you can have a fillet that kind of just tears apart altogether. Most cases, it can be just on the fringes or on that kind of more like this first picture, kind of a soft area in that cranial region of the fillet. So Dr. Shai and his colleagues in Canada did a recent study, came out in 2022. And I like this graphic that they show because I get this question a lot about how much overlap are these myopathies of white striping, woody breast, and spaghetti breast. So basically, he's about almost 10,000 birds, and about a third of all the fillets that they surveyed had all three of the myopathies together. About half of those have woody breast and white striping together, and then they saw a little bit of a seasonal effect. White striping could be there with no other defects, a little bit more prevalence of that alone. But a lot of our fillets, again, have some sort of one of these defects associated with it. Again, that kind of goes to, hey, we've got the potential for it, and how do we manage that through the process? So detection's one aspect. This is just a list of some of the things that have been studied and more times, and I've got a few more slides on this, so I'm going to quickly go through that list. I'm going to talk about really the ones that are starred. The standard method of detection, and we're talking really about woody breast and maybe spaghetti breast too, but is to palpate that meat. It's really hard to the touch, and it has a varying degrees of severity, so very flexible to very hard throughout the muscle, and we can hand palpate that for detection, but as you might expect, that takes a lot of labor and training in the plants to do that on a regular basis. And so if we want to try to automate things, then we want to have a good way to detect. Using instrumental compression force is a good way to instrumentally and be a little less subjective. It is highly correlated to our hand scoring that we do. And I've got a little picture of a robot. I'm going to talk about robotics in just a minute, but you can, there would be potential to use a robotic arm to kind of look at the compression force associated with fillets if that were needed. Near-infrared spectrophotos, whatever, I can't say that this morning. So NIR is used in plants. So there is a system called TOMRA, and it is used to assess muscle composition. So woody breast tends to have lower protein, higher moisture, and so that's in a couple of plants. And I was just in a plant recently where they were using it, and they did a good job. The fillets that they were identifying and kicking off to another area that could be used in other products, like a ground product, for example, then everything that was being kicked off was more the severe woody breast. So you kind of have to set that ranges up per plant and what your requirements are and how much you can kick off and what's important in determining. It also goes with your product mix. And what you need to sort out, really. Hyperspectral imaging is really being used, and a lot of research being done in this area. So it's just NIR with digital imaging. So they're looking at this for woody breast detection. I know there's some in the audience that have used this as well. There is one unit that's being tested out there by PP Optica. And another added benefit of hyperspectral is that they could look for foreign objects as well, which would be hazards. USDA in Athens, they have looked at a side view machine vision system to look at the muscle rigidity of the fillets, and they can use that imaging technique after the fillet is coming off of a belt, how stiff that fillet is versus how flexible. And they can look at the bend angle or the bend energy associated with that to detect that woody breast. And so I think some of the ongoing research with them is to combine this with maybe other techniques like hyperspectral imaging and so forth, but they do have a patent on that imaging system. We did a lot of work to look at image analysis to detect woody breasts on a carcass standpoint, and so we know there's vision systems out in processing plants. We wanted to see if there were shape features associated with woody breast. And so we determined that there are. We do have a patent on this, but it's not incorporated into plants at this time. But and you say, well, it's at a carcass level. Well, I see the value of this is to give us data. So there's a lot of data, as the previous speaker talked about, that we could work with. If you know that, hey, there's a trend, like certain flocks have a higher percentage of woody breast than other flocks. Okay, what are the characteristics of those flocks versus the ones that don't have it? Or, hey, I've got more product that's going to be coming down the line. Let's be ready for it. So other areas of research and development and processing would really just be the processing aspects, and I've kind of lumped a lot of things in here. But automation and innovation, robotics, big data. I'm not going to really talk about big data a lot, but there's a few in the audience that could probably handle some of those questions. But robotics and processing, so again, we have labor shortages. This kind of creates headaches for employees or employers, and their HR groups. So those can have, they're dealing with people calling in sick or quitting or whatnot. We're dealing with ergonomics and we're dealing with food safety. All of those things can lead to the need for more automation. Some examples that robots or robotic usage have been in like packaging processing or moving products. So last year it was announced that there was the Tyson Ventures invests in an AI-powered robotic system. So they have like this gripper to move their product. And if you were in Atlanta this year, you probably saw a lot of that in Atlanta, these gripper type objects where they can pick and place product. Deboning and intelligent cutting. So this is another robot that was in the literature in 2016 where they called it a gribot. I'm not really sure how you pronounce that, but used a 3D image vision guiding system to debone that was out of Norway, I believe. But you can use this for quality measures. Like I said, that robotic arm where we can measure hardness is one thing, and then maybe automated sanitation processes. So intelligent cutting, just to say a few more things, Georgia Tech Research Institute does a really great job of some of their really cool processing techniques. I really encourage y'all to reach out to some of those that are here today in that group. But they have been working on intelligent cutting, so using 3D imaging. They use machine learning, those statistical models, all that big data that's kind of beyond me, but there's some robotic arms in place. And the goal in this, I think in the early stages, are really to work on that initial cut. So the initial wing cut is really important. And if you do that wrong at the beginning, I tell my students that if you mess up that initial wing cut, then the rest of the product is going to go to give you some variation in your data later on. But it also can help prepare that wing and or for breast deboning. So doing that cut and then sending it on to a debone process. Virtual reality is another cool kind of innovation that's being applied in a lot of different fields in science. Used it now, since I've been saying it, it's popping up on my Facebook now that we're using virtual reality to do operations on people. I'm like, okay, I'm going to show my daughter that. But anyway, virtual reality and robotics, there's a place of that in poultry. So again, the Georgia Tech Research Institute is doing some work on a variety of things, looking at cutting trajectories using virtual reality of the VR systems, pick and place objects, testing new technologies. Really, that's all I'm going to say about that, other than Colin Usher is kind of leading that group. I'm not sure if he's in the audience today, but he is giving a presentation this week on Thursday at 11:15, and that's abstract 259. So I encourage you all to go see that. If we're lucky, there might be a demo, I've heard, but I could be wrong. If he's in the audience, he's like, no, I'm not doing a demo. Anyway, moving on, chilling technologies. There's some new chilling technologies that are being looked at. Sub-zero chilling, ice water slurry, which is kind of the same concept, but also using rotational shackles. Some of these have been developed by Georgia Tech Research Institute as well, but the application of this is really to improve that chill efficiency. If you think about our birds that we're processing today, they're real large. It takes more time to chill those carcasses. So anything we can do to help decrease that dwell time can help production and so forth. But it can also improve food safety. So Dr. Kong is in the audience somewhere. He's been working on sub-zero saline chilling. Just increases that saline concentration to decrease your water temperature. Obviously, they found that using 4% sodium chloride at minus 2.41 Celsius combined, they've done some research with the hot water spray also, and using those things in combination has given them a bacterial reduction. So it gives you some improvement in food safety and also that decreasing dwell time in the chiller. And so some of the questions as we go forward is, hey, if some of these chilling technologies can improve that bacterial reduction, can we reduce the use of antimicrobials or eliminate those? Rotational shackles and ice slurry chilling, again, this one's from Georgia Tech Research Institute. They're looking at rotational shackles in the chiller. And by doing that, we have agitation in the chiller, but it can help move that water around, and combined with the ice slurry can help reduce some of the need for that the antimicrobials and so forth. More research is needed in this area, especially for the large bird aspects, to see how much a reduction in that dwell time and bacteria that we can get. All right, just a couple more slides here from food safety aspect, and I'm not a microbiologist, so I just try to work with great ones when I can. But food safety, obviously there's microorganisms involved, but there's also hazards like foreign material. So the use of antimicrobials and processes can lead into that research. There also is a need of quantification of pathogens rather than presence or absence in the processing plants. We need some baseline data on that and what it all means, Saravar testing, and then that would also then lead into policy revisions, because right now USDA requires just that presence or absence, and what does that really mean in terms of human health? So the future outlook for food safety is to use that multi-hurdle, multi-technology approach to food safety. Can we use the use of novel antimicrobials? So we're using a lot of PAA right now, but there are other antimicrobials out there. We're not saying that we need to ditch one, but maybe we add some to the mix in different spots where you get that multiple hurdle approach. So bromine's a product, bacteriophages are products, that, it's a virus that will attack a bacteria. But all those can be used together to help get you those bacterial reductions that are necessary. So continue research on microbiome. How does the microbiome, how is that impacted by the interventions that we're using? Bacteria are, I say, smart. They can adapt to things. And so understanding those things and how the bacteria are reacting are important. And then finally, the precision use of antimicrobials. If we are doing all the Saravar testing, can we develop these novel antimicrobials for targeted use in certain applications to really target those microbes that are of higher concern? All right, this is the last one, and my final thoughts, but the food safety and processing innovations, which I've already mentioned these, is that robotics and automation, can we handle that food product, like the gripper, for example, or the sanitation aspects. Vision systems, hyperspectral imaging for foreign matter detection, and then the chilling technologies. So, to conclude, my final thoughts would be that our innovations, I think, will continue in the area of poultry processing, and this area is important, including that use of artificial intelligence. So there is a lot of big data out there that we can take advantage of. The live production impacts, I've got nutritional genetic management. All of those things on meat quality and safety are going to be important as we move forward, all right? Understanding our quality issues and developing mitigation strategies are really a must, and to prevent those defects and minimize the effects. You know, once the processing plant gets it, how do they deal with it in the short term? And then again, food safety is always going to be important, and developing those new methods for pathogen reduction will continue. So with that, I know y'all are ready for a break, but thank you for attending, and I'll see you in a bit.
Thank you so much, Dr. Owens, for the great and educational overview and outlook. We're now on a coffee break, and we will be back at 10:30.